Welcome to another Foldit Lab Report. I am BCAP, here with my colleague Ian H. at the Institute for Protein Design here in Seattle. If this is your first time discovering these Foldit Lab Reports, we produce these videos on the first of every month to tell you more about the science that goes on behind Foldit. The big news this month is Foldit got a makeover. We have completely reworked the user interface, which is what you see when you open up a Foldit puzzle in the game. Experienced players will notice that we've moved different elements of the interface around. This should make it easier for new players to find important tools in the game. This change also makes it way easier to customize how molecules are displayed on screen. To unpack this big update, we have something a little bit different for this video. With us today are two members of the Foldit team from Vanderbilt University in Nashville, Tennessee. These two led the development of this brand new interface as well as the small molecule updates that we talked about in the last lab report. Link in the description. Welcome Rocco Moretti and Siren from the Milo Lab. So what's behind this big change in the way that Foldit looks? What was wrong with the previous original and selection interfaces in Foldit?
Yeah, so one of the biggest challenges that we have as Foldit developers is having to develop new tools and new visualizations in respect to these two interfaces. This doubles the development time and makes it extremely difficult for us to track down bugs that either are existing or that may come up with new implementation. Consolidating these two interfaces will help us address problems much faster than we have been able to in the past. Another issue that we see having two interfaces is new players coming into the game experience the tutorials in one interface and then get help later on from experienced players in a different interface. And this causes a bit of a disconnect in their learning of how to actually play the game. All in all, the unified interface has concrete advantages for Foldit as a scientific project.
I'm personally excited about the new view presets options. It means when scientists like me create a new electron density puzzle, I can launch it with the electron density view options already in place. Or if I'm posting a protein design puzzle, I can make the options right for that too. How do the presets work from the user side? How will players benefit from this?
Yeah, so this is an exciting new feature. And with it, customization really is the name of the game here. So what users can do is they can pre-select all of their view options that they want for the puzzle that they're tackling at the moment, and they can save that view option for later. In the presets menu, the player can toggle between different presets that they've saved and also that scientists have put in the game for them that we really think will actually help them along the way.
Let's go to our puzzle updates. Rocco Moretti, what can you tell us about the latest small molecule design puzzles?
So Foldit players have been really good with coming up with novel ideas for protein folding and protein design. And so the point of the small molecule puzzles is to see if we can use that ingenuity that Foldit players have in creating new small molecules. And small molecules are rather important because a large number of drugs, particularly the pills that you take as drugs, are based off of small molecules. For our initial drug design puzzles, we're focusing in on a class of drugs called PROTACs. PROTACs are an innovative new strategy for targeted protein degradation. This means that we can select particular problematic proteins in the body and then remove them. One interesting feature of PROTACs is that they're sort of split up into two parts. One of them targets that protein of interest that you want to remove, and the other targets the cellular degradation machinery. For our small molecule design puzzles, we're not creating an entire PROTAC. Instead, we're focusing in on the portion of the PROTAC that binds to the cellular degradation machinery, specifically the protein VHL. With good VHL binders, that will make it easier for future researchers to create PROTACs to their specific molecule of interest. One of the exciting things about the small molecule design puzzle series is that our collaborators, Boehringer Ingelheim, have committed to synthesizing promising compounds that have passed all the filters. And that brings us to the design of the month. This design is by an anonymous user from puzzle 2068, VHL ligand design round 2. The goal of round 2 is to change the central hydroxyproline of the ligand. This anonymous user didn't change very much about this eastern end, this biaryl group. Instead, they concentrated mainly on this central region, which is one of the reasons we like this molecule. We like the fact that this hydroxyl has been maintained from the original structure. This hydroxyl is important because it's making two hydrogen bonds to the protein. If we don't have this hydroxyl or something equivalent to it, both of these atoms that are on the protein would become unsatisfied without a hydrogen bonding partner. And just like bonds are bad in protein-protein interactions, they're also bad in protein-ligand interactions. So this hydroxyl is still present, and it's still attached to a 5-membered ring. But this 5-membered ring is changed up from what it was, so it's no longer a hydroxyproline ring with a nitrogen. It's now entirely a carbon ring. What's also very interesting is it's now a fused ring system with this aromatic ring, this alternating double and single bonds. This fused ring system is one of the things we like about this design. One thing that might be a little bit concerning is these two nitrogens that are double bonded together. I'm not sure how frequent those are in drug-like molecules, but one of the things we can do to check to see if this is an issue is to take a look at the objectives. In drug design, there's a number of important factors that go beyond just the how well does this small molecule bind to the protein. And the objectives try to account for these sorts of things. Significantly, there are a number of factors that play into how well the molecule acts in the body before it even gets to the protein of interest that it's binding to. One of the big considerations that we have is the efflux of these molecules. That is, how the body handles them and removes them from the cells. And two of the big considerations there are both the hydrogen bond donors as well as the TPSA, or the topological polar surface area. So both of these considerations mean that the efflux is not exactly what we're looking for as for a drug candidate for the starting molecule. And so keeping an eye on these means that the compounds will be better for actual drug purposes. And it does look like this particular design does well on both of those considerations. The anonymous designer of this compound did a sufficient job of reducing the polarity. However, this compound might have gone a little bit too far in the other direction, increasing the C log P, or the hydrophobicity of the molecule. The C log P is a little bit higher than what we would like, and that ends up reflecting itself in not getting the full C log P bonus. So this molecule is a little bit more hydrophobic than we would like it to be. That might be solved by adding some more polar groups, but again, if you add too many polar groups, then the TPSA and the hydrogen bond donors have a potential for increasing. And small molecule design, even for the professionals, is this very delicate balancing act of trying to optimize one property while not compromising the other properties. So all in all, this is quite a nice design from the anonymous player. One thing we like about the molecule is how the center has been changed up a bit, while still maintaining those key interactions that allow the compound to bind tightly to the protein. Having a diversity of design and a range of ideas is really one of the strengths of Foldit, where we can get novel and innovative approaches towards these sorts of molecular design puzzles.
Thanks again to R. Moretti and Siren. That's all for this month. As always, thanks for watching, thanks for playing, and we'll see you next time.